I'm going to introduce my class about advanced methods in analysis and in computing with application to modeling microdomains. Especially I will focus to two main specific domains, which are the cell nucleus and synaptic terminals. So this is the classes that will occur this year, 2013-2014. It will be at École Normale Supérieure in Paris, and if you are interested, you can look at all the information in the following website. So the overview. In this introduction, I'm going to present a general notion about the cell, about neurons, and what is cell compartment, and also specifically about the cell nucleus. Then I would like to discuss more specifically physical modeling, how we go from the biology to the modeling. Then the third part would be about how to develop mathematical methods. So what I have in mind by mathematical methods, it's going to be how we do asymptotics on partial differential equation. How do we study specifically stochastic processes, which are usually dynamical system perturbed by a small noise. An important part is also to design rational simulation, especially Brownian simulation, that account for what happened inside the cell. What I have specifically in mind is stochastic chemical reactions. And finally, an interesting consequence is on data analysis and how to extract parameter and features from data. And I would like to emphasize how this mathematical method and simulation allow specifically to do this. So just to remind you the basic concept of what is an eukaryotic cell, you have a cell in here, the nucleus is inside where the genetic material is located, and you have here the cytoplasm. This region is quite interesting because a lot of molecules have to traffic inside this position. Let's say when they are synthesized at a point here, molecules have to travel to be anchored for those who have to be located on the membrane here to occupy their position. An interesting and still open question is how the nucleus, how the DNA is organized in the nucleus. So, to give you a more specific example of a problem we will be interested in is, suppose you are interested in a virus. You put a virus particle that has to move inside the cytoplasm. How the underlying microtubule network is important in defining the trajectory of the virus that has, for example, to reach here a small nuclear pore to deliver his gene. So how to account for such organization of microtubule in the motion? This is a type of question that we would like to formulate more specifically in mathematical terms with stochastic processes. So now you have here a picture of the nucleus. You have here a small nuclear pore, and you can see that there are basically two regions. The region where you have large condensation of the chromatin in here, and here you have more space that is considered to be more empty, much less dense than the gray ones. So this is the dense region in here, and this is the less dense region. So how to account for such diversity in the nucleus? Another type of cell that we are going to be also studying is hippocampal neurons. So you have here an example of a neuron with the soma of the neuron, large dendrite large axons in here. And here you have all the dendrites that can be spread to other neurons. So one of the questions we'll be interested in is how neurons are sending information and how to model this, specifically to what happened at synapses. So to better understand this notion of synapses, let's take the example of the retina. So the retina is basically a piece of the brain located at the end inside our eye where the light is coming in. So the light goes until it hits here specific cells, which are called photoreceptors. There are two types of cells, the rods and the cones. So you have a photon hitting here the outer segment, and you have here a representation where a photon can be absorbed, which means activate a rhodopsin molecule, and this can generate a cascade of chemical reaction that will lead to a cell hyperpolarization. So those are rods for night vision, cones associated to daily vision. It's still not completely understood what's the role of their specific geometry and what's the consequence to how they can modulate or constrain the chemical reaction that occur in these microdomains. So once the information has been leads to a hyperpolarization of the cell, it's actually the next stage is to see how this information is transmitted at the level of synapses, which are highlighted here. And so there are different types of synapses that are all over the place. And the information can be then transmitted from one layer, which is here the photoreceptor layer, to the ultimate layer, the third layer. There is another intermediate layer here where some processing occur. But this layer is interesting because this is what is sent to the brain. This is where ganglion cells are located. And then it goes directly to the brain. So synapses is a very common feature. Actually, if you just start with the following calculation, that there are 10 to the power 11 neurons in the brain, and there is about 10 to the power 3 synapses per neuron, then you can imagine that we have 10 to the power 14 synapses in our brain, which is quite a lot. So it's still very interesting to understand how they are working based on their molecular component. So more specifically, so you have here an example of a neuron. So once some local depolarization occurs at the synapse, in some sense it is integrated somewhere here, and it can lead here to an action potential that spread and can lead here to the release of neurotransmitters. So there is also an interesting features which are called dendritic spine, which is this small protrusion that you can see here all over the place. And so this is also a microdomain of great interest, because today we still do not fully understand how they work. They have an intermediate shape. They are at an intermediate size because they are of the order of, this is about 1 micrometer. And this size is very particular because they are using really a few numbers of receptors, which is really at the discrete level. And they can integrate information so that the information is integrated in the dendrite. So the question we will be interested in is what's the function of these particular microdomains. So how to characterize neurons? The best way to characterize them is to use their electrical activity. If you just stick an electrode inside a neuron and you record the activity, you will see, for example, that there is a resting potential. That is, if this is the voltage of the neuron, they are usually at -60 millivolts. This is called the resting potential. But actually this is not entirely correct, because as you can see here, the voltage is actually changing between a low resting state here, which is called a down state, to another state which is called an up state, what you can see here. So the voltage potential can switch between those two states, leading to some complicated behavior. So you have here an example of a distribution of the time spent in the up and down state. Interestingly, if you give cocaine to the neuron, you see that this activity is completely abolished, and this is an irreversible process. So classically, neurons can have sophisticated behavior, and this is actually seen in the process of synaptic transmission and plasticity. We have seen that if you stimulate with this electrode a neuron, and you record for another neuron from which the link between the two are just through this micro contact, which are called synapses, then you can see that such a stimulation in here can lead, if you record here, to such excitatory postsynaptic potential, which is a local depolarization here that can spread enough. And under a protocol which is called long-term potentiation, you can see that the next time you provide a stimulation at the first electrode number 1, when you record at electrode number 2, instead of having this response, you obtain that response. And that response can last for hours. And this phenomenon is called long-term potentiation, and it's underlying learning and memory. And you have also the possibility to revert it, which is called long-term depression, and you can reverse it. You can go from 2 to 1 in here, and this can last again for hours. So we will be interested in how can we calculate this current from a molecular event and their biophysical property using a specific modeling about synapses, and especially what happened here in the synaptic cleft. So why studying synaptic transmission? I would be brief, and I would say that we are still missing the molecular details of this synaptic transmission and their modulation. So it will be interesting once you know the player to just put them inside a rational simulation, so that we can see precisely what they contribute to synaptic transmission. So for example here, a receptor can be released, moved by Brownian motion, until you hit here a receptor from which a current can flow inside here, the postsynaptic density. Of course, synapses underlying the network property, so if we understand specifically and carefully the synapse, we might be able to understand the network properties and how the synapses underlie higher brain function. Interestingly, it is not possible to link molecular event to pathology. So let's take, for example, autism. Autism has been found recently to be due to an alteration an alteration of molecule, leading to a reorganization of the presynaptic versus the postsynaptic, and specifically some protein are affected, which are called scaffolding protein, which are located here in this region. So if we know that some of the molecule of this protein, for example, are not functional, or their position is changed, we can test their effect in terms of in a rational simulation. And finally, to give another example where now it's possible to link a pathology to the molecular detail, is a case of epilepsy, where it is known that the ketogenic diet, which mimics starvation, can prevent the seizure, and this is due to the following effect. You have here vesicles, and vesicles that contain a certain number of neurotransmitters actually in that case contain less neurotransmitters. So the number of molecules that are going to be released here is much less than in the normal case. So what is a synapse is basically composed of a presynaptic terminal that is shown in here and a postsynaptic terminal. And the dimension you should have in mind is this is about 1 micrometer. And the number of molecules that are located here are the order of, let's say, 5, 10, let's say to 50. So it's not a lot. And so if we are interested in making a model, we have really to use a stochastic description. So let's now continue with this dendritic spine. So you see that they have really a particular type of shape, from philopodia to more mushroom type spine, and you can have up to 300 thousands of spines in a single neuron. So what are they good for? This is still a mystery. Now I would like to go to another scale, again to show you another type of modeling. So this is about neurons that can form small connected network. So you have here an example of neuron that connects to 5 to 30 neurons connected together, and this can lead to very interesting properties. So for example, if you stick an electrode and stimulate here a single neuron, then for about, let's say, 5 milliseconds, all the network, all the neurons together can lead to a response that actually can last something like of the order of 2 seconds. And these show the different colors show the calcium inside the different cells. So these show a synchronous activity of all the neurons together. So what's the reason for this and how to model this? So we will see that we can use some mean field model to precisely account for this phenomenon. So now I would like to give some more specific definition. So what we call by microdomain? This is going to be part of a cell that is driven by molecular interaction that underlie a specific physiological functions, something that is defined in terms of the response. So this is the example is if you have a synapse is the structure that mediates the information between pre and post neuron. And still, you have here a specific microdomain that defines it. So the outer segment of the photoreceptor is another example where the goal of this is to transduce a photon arriving into a cell hyperpolarization. So why modeling microstructure? Of course, we would like to understand what they are doing. And they are usually at the limit of the time and space resolution. So the function of microdomain underline many cell function, and we want to analyze them to understand their behavior in normal and pathological condition. We would like to account for this in our approach to the small size of the structure, the fact that it has a low number of molecules, and this low number of molecules is such that if you introduce external dyes to visual them, you can perturb all the cascade of chemical reaction. Thus, modeling here has a very interesting role. So we would like to use this to study how a molecular cascade is really constrained by the geometry. This can lead us to predict the effect of adding drugs or removing specific molecules. Now the role of modeling is not only we would like to derive some physiological function, but we would like to do this using the basic statistical physics principle. So would be interested in computing various biophysical properties and obtain the dependency with parameters. And this can be done using asymptotic formula and that can be obtained from mathematical analysis. So we have already said we would like to reconstruct the intact function and to study the effect of the knockout or the effect of adding specific drugs. So the question we will be interested in, although they are a question in biology, I think we could address them in modeling term. So how cell maintain and regulate protein. So if you have a protein that is synthesized here and has to move to here, how it finds such a target and what's the time to find it. And in more general, how structure defines function, which is well known for channels, is much less understood at the level of a cell. And of course, the question of how many molecules, how long it takes, could be answered if we can formulate the associated equations. So what's the goal of the class? Derive physical models from the from the biological from the biology. We like to analyze the model equation, and this is usually solving partial differential equation of stochastic processes. Let me give you an example. If we're interested in the mean first passage time, we know that the mean first passage time satisfies a partial differential equation, which is epsilon u plus b gradient u, where b is a vector field, equals -1. And so we would be interested in looking at the solution of such equation as epsilon goes to 0. So how to calculate asymptotically what u is. So we will present methods for asymptotic calculation, and we will specifically spend time to analyze the diffusion and chemical reaction. So this can be, for example, the small hole problem, how long it takes for a particle to escape a domain through a small hole when it arrives here, or the chemical reaction, which is, for example, if you have many different binding sites, and you can have inside, let's say, molecule that move by diffusion, and when they move, they can hit one of these sites so that you can have a molecule. So, for example, you can look at the following question: what is the number of sites that contains this interaction? How long you have to wait, let's say, to have 5 sites that are occupied? So how long it takes for this to happen. All right. So we would like also to design rational stochastic simulation. How do you do a simulation, let's say, for example, of the particles that has to be partially absorbed here? That is, sometimes it can go through and sometimes it is reflected. Or another question is, suppose you have a cooperativity, you have one particle that moves by diffusion, it hits here, but then it needs another one to come in order for this one to go inside. So how do you model this? This should go away, or how long this has to stay in this specific location. And from this, we will be very much interested in estimating statistical quantity, as we said, the number of bonds, the concentration of molecule in specific condition, and also to analyze data and to extract data from electrophysiological recording or from super resolution imaging. So we will spend a bit of time discussing this type of procedure. As we said, the output will be to better understand the microdomains and to quantify the role of the geometry, computer properties, and to predict the organization and to understand the role of key parameters. So now let me discuss a bit the syllabus, which is the decomposition of the classes. So basically we'll have the first part is going to be mathematical modeling. So we'll start with the Brownian motion. I'll show a stochastic approach to understand the stochastic equation and the link with partial differential equation. So the link it will be done through the Ito calculus. I will spend quite a bit of time on the diffusion equation, which is dp dt equals the diffusion Laplacian p. This is for the probability density function. We'll spend also some time on the Fokker-Planck equation, which is the equation for the probability density function for a general stochastic process x dot equals b of x plus sigma w dot. So I will explain what's the noise and what is the meaning of this stochastic equation. So this is a drift and this is noise and this is the velocity. And then we will spend some time on describing the motion of a polymer, especially of a monomer inside the polymer. Then I will spend time on mean first passage time equation. I will derive what they are, and especially looking also at the distribution of exit point. So the most important example is the escape from an attractor, where you have an attractor point here, where all trajectories are converging to that point. And so the question is, of course, how long it takes for a trajectory to escape when it is perturbed by noise. And of course the question is where it escapes. What is the density of escape point? So I'll also spend some time on the narrow escape and small hole theory, how long it takes to escape through one hole, how long escape also to escape when you have two holes, 1 and 2, and specifically what happens when you can change here the distance d between the hole. I'll further discuss diastrate time, which is the time it takes for a particle to enter into a cusp here, which is much harder than just to find something located on the surface. And for this we will have to use the theory of conformal mapping. I will also spend some time on defining what are Green's function. So then the most important part of the class will be to study the singular perturbation analysis, in particular what are the methods to solve a partial differential equation. So we'll discuss mixed boundary value problem. Again, we'll show how to solve the small hole problem. After that, we'll have given here the equation. This depends on the understanding of boundary layer techniques, which are very important. I'll give some example of how to calculate the spectrum of non-self-adjoint operator. This with the specific interest of the Fokker-Planck, the distribution of exit time, and application in neurobiology. Then we will continue with modeling shape objects, and here this is really where I want to discuss the Ross polymer, and specifically when you have a polymer which is an ensemble of beads connected by spring, so bead k, k plus 1, k minus 1. What's the motion for these beads? And how can we study, for example, the time it takes for a polymer to form a loop, that is for the end beads to come in a close vicinity, let's say, of in a sphere of radius epsilon. Then we will focus on the theory of Markov chain to describe chemical reaction, and we will use the narrow escape to define what is the forward rate, use the backward rate, and then define, and I will explain what is the threshold theory, which is what we just discussed before, how long it takes if you have several sites for, let's say, the probability or the mean time for the site to contain one relevant molecule that can interact. Finally, we'll have, so this is, all of this can be considered really as the main mathematical parts. And now for the modeling aspect, we will go to two specific examples: synaptic transmission. I'll discuss receptor trafficking, how we study diffusion in narrow domains, that is, suppose you have a synaptic cleft. How do we study diffusion in here? How we define the synaptic current, the residence time of a receptor in the synapse, diffusionoid dendritic spine. So suppose you have a spine here, how long it takes for an ion to escape here from such a structure. And this residence time is important because it can define the possibility to have plastic changes that will affect the geometry of the spine. We'll also discuss electrodiffusion. I will present mean field model for synaptic depression and some application to understand some neural network. We'll present some model of synaptic transmission and in the process of up and down state, how can we compute the distribution of time in such a state. So finally, we'll discuss the nucleus organization. I would like to split it into 3, A, B, and C. So first is going to be about modeling the telomere lengths. So for this, we will use a Markov jump process because telomere can be seen. This is the length of the part of the end of the chromosome between cell the generation n and generation n+1. Either the chromosome is shortened, that is, a piece is removed, or it can be lengthened. So if you have such a possibility at each cell division, then you may ask what's the distribution of telomere lengths in the end, and especially what happened for the shortest telomere. So this can be done using Takachik equation, which has been used in queuing theory. So the next thing I'm going to discuss is about telomere organization. It's so how telomere are organized inside the nucleus, and we'll use the theory of aggregation dissociation of particles inside the bounded domain. So the final part will be the search process in confined environment, and specifically how, for example, if you have a DNA, how the DNA loop, how long it takes for DNA to form a loop. We'll also be focusing on double-strand DNA break. So if you have a DNA and it is broken, how long it takes for the two pieces to glue again together. And more specifically, how we can use our analysis to understand the motion of a single locus, how we can extract information from live cells imaging. All right, so this is more or less it for the this introduction. So you can find more information in our website. Some requirements are some ideas of what are partial differential equation and their analysis, some notion of probability, of Laplace methods, which I may do a specific class on this, and of course some notion of cellular biology. The evaluation will be small project. Additional reading for the class are construction, for example, of the Brownian motion, some idea of what's the theory of probability, some notion of partial differential equation and differential geometry. Dynamical system, I think it's very important to understand, the basis of a recurrent set. If you have a generic hyperbolic dynamical system, x dot equal b of x, and of course some notion of biology. I hope that some of you knows how to make some simulation, either deterministic or stochastic. A tool can be the FreeFEM for solving a partial differential equation. So further background, the Pasteur Institute offer classes that invite you to go and listen. You can find some classes in statistical physics at École Normale here in Paris, and at Paris 6 in Paris Savin University in Paris, in Orsay. The Current Institute offers also a variety of classes, as well as Gustave Roussy Institute, and this is more directed to cellular biology and for cancer research. So some references. We have just recently published this following review, which I think give you a pretty clear idea of how analysis can be applied to physical for biological domains and cell biology in general. This is a more specific article about Brownian motion in specific microdomains. The theory of narrow escape has been described in our early work of 2006. Threshold of activation for chemical reaction can be found in this publication with Kandau-Duc. And in terms of the book, I think a good summary of what is going to be presented here, although it's not covered, but to have the essence of some proof and other references, I invite you to really look at the book of Zev Schuss of Theory and Application of Stochastic Processes. A good book for reference in biology is the Classical Molecular Biology of the Cell. You can also have a look in the book of Schuss of 1980 for the theory and application of stochastic differential equation. For partial differential equation, the classical book of Garabedian will be enough for us, especially for the description of Green's function. So now we are ready for the next class, which is going to be class number 1. So I hope that with this introduction, we'll be able to see where we are going, and I'll be happy to have you for the first class. Thank you.